It's my pleasure to introduce Professor He. He is currently an assistant professor at the Institute of Molecular Medicine at Peking University, and also a joint at Peking Tsinghua University. And he's interested in gene regulations and cell behavior of tissue formation and regeneration. And he also developed a lot of interesting technologies like Singlecell, ChipSeq, and etc. And with that, I would like to pass to Professor He, please.
Okay, thank you. Thank you for the introduction. And also thank Guoji and all organizers for organizing such a wonderful cyber conference. So today I'm going to show you our recent progress developing a new method for single-cell joint detection, chromatin, occupancy, and transcriptome. So exciting with the ability to reconstruction a high-dimensional epigenome. So single-cell RNA has been progressively used for catalog cell type and understanding the cell heterogeneity. So with the advanced in single-cell epigenomic technology, we're able to investigate the genome regulation and the mechanism in sulfate decisions. Actually, the gene activity was largely embedded in the epigenome, so including the selective transcription factor binding and what started the chromatin accessibility by ataxic and various histone modifications as well as the DNA modifications. And it's also known the transcriptome and epigenome can affect one another to separate transcriptional homeostasis and establishing sulfate. So that's the prior in the single-cell multimodal omics will be very informative for reconstruction of gene regulatory network, which is driving the specific cell identity. And so think about the pairing single-cell omics across different modalities, such as RNA-seq, RNA, and multidimensional epigenome landscape. So you have two choices. The first one is the computational method. However, inevitably might lead to inaccurate interpretation of biological meanings. The second is becoming possible to experimentally measure the several omics in parallel in same cell. Actually, there's many promising progress made toward this goal recently. For example, three methods recently reported for single-cell joint assay of chromatin accessibility and transcription, such as SCL from Jason Durenab and Singlecell from Quinn Zhang and PALSIC from Bin Zhen at UCSD. So, however, development of technology for joint measuring chromatin accessibility and the transcription is far behind in the field. So our lab striving to fill this gap. We recently reported two single-cell chip methods. So one is the co-batch, we call it co-batch, with the core design and as in Carter Taylor, independent reported by Steve Henikoff lab. And another one I to show you is recently also published last year, single-cell IT chip. So this one with unique advantages starting from tens to a few hundreds of single-cell, and also with an advantage for the library construction to avoid the low-risk custom sequences, not only reduce the cost, but also widen adoption by general laboratory. So motivated by those two single-cell methods, we developed designed the COTEC and for single joint measurement of chromatin occupancy and transcriptome by simply combine with modified SMART2, just look at the 3' end of mRNA. So after many attempts in Jie and Qianghao made some progress. So on the left panel, the species mixture experiment with NHCT3 and human cell line translate T cell showed K4 chimerization methylation rates and RNA-seq from human and healthy cell line are uniquely mapped to the human with the color of 85%, unfavorably mapped to the mouse genome. So with this cutoff, the collision rate about 4%, and which is acceptable for the downstream analysis. So in the middle panel, the aggregate COTAC H3K27 chimerization single are enriched at the typical marker Raman gene, which is also observed highly enriched in bulk data. And so what peaks from three groups are well aligned, suggesting the reproducibility for the single identification. And on the right, the scatterplot showing the robust data reproducibility between two independent experiments. So not only on the H3K4 chimerization, and also on the transcription. So when culture on the serum medium, mouse embryonic cell, embryonic stem cell represent a rather heterogeneity with naive and primed pluripotent state. So we performed COTAC on H3K27 chimerization and H3K4 chimerization, and classed the single cell together. So on the left panel, inspections the single on a specific loci, such as highly strongly expressed OX4, enriched for the H3K4 chimerization, and vice versa for the silenced gene GET2, enriched for the K27 chimerization, depleted for H3K4 chimerization. So on the top right, single cell were separated into two classes by COTAC RNA-seq data, C1 and C2 corresponding to the naive and the primed pluripotent state respectively. And so all single cell from the different experiments, K4 chimerization and the K27 chimerization are well matched, indicating the less batch effect. So similarly, also performed clustering using the H3K4 chimerization data and identify two clusters, C1 and C2. And so when we map it to the DNA data to the RNA data, so what cells within the same cluster from both DNA and RNA are labeled as matching, otherwise discordant. So the matching rate about 68.7. However, when we do the same thing on H3K27 chimerization COTAC data, identify two clusters, however, it come up with a much higher matching rate, about 82.6. So that suggested that H3K27 chimerization data represent a much higher cell type or state specificity than that in H3K4 chimerization. So let's to demonstrate the ability to COTAC in a multidimensional epigenomic reconstruction. So I attempt to assemble two histone mark within a single cell to study the gene-regulatory relationship. And one was most interesting in the epigenetic field is the bivalent chromatin domain, which identified about 10 years ago. So which are they are co-occupied by the active histone modification H3K4 chimerization and the repressive H3K27 chimerization. So previous bulk chip data by those two marks supporting the free model, and however, recent data from the ReChip and the other experiments that this overturned this model. This model just suggests as not existing bivalence just due to the low resolution in the bulk chip data. Instead, with a nice experiment from by single cell nucleosome imaging from Bradley Bernstein at Mass General Hospital, supporting about a vast majority present in the model B it's K4, K27 histone modification on the same nucleosome. Very few, about 4% in the model supporting the model C, is the co-occurrence on same histone, the same histone. So, however, the question is the spatial genome resolution is still lacking. And so we're trying to map the histone mark occurrence at the single cell occurrence at a single cell resolution, and we asked whether we could harness the COTAC data for genome-wide mapping and bivalence in this case. So this one is not trivial because it not only permit construction high-dimensional epigenome landscape, but also allow us to uncover the regulatory relationship between different molecular layers. So to have the simplified workflow, where you perform the COTAC on different histone mark and class the single cell by the transcriptome. And ideally, you could obtain the pseudo single cell for the further subclustering. So in this case, it's 3K4 and K27 trimethylation. So with that on the right, we could calculate the bivalency score, and with this interpretation, identifying the 3 classical groups of genes, active and bivalent for priming and the recursive gene. So we identified the 3 groups according to the dynamic bivalency score along the pseudoterm, class 1, with a decreased bivalency score along the pseudoterm. Class 2, the peak in the middle, and the class 3 exhibited a gradual increase. So expected all of genes was associated with lower expression in that stage. So also can be exemplified as several transcription factors, such as NK222 with decreasing the bivalency score, and also the decreasing K4 and K27 trimethylation signal, the same to the other 2 transcription factors. So, so in the second part, I'd like to show you how could we harness the COTAC to study the gene regulation for sulfate bowels in a context of biological process, the blood development. So we know that 3 waves of hematopoietic cell formation, but only one, the last one, generating the long-term self-renewal hematopoietic stem cell with full potential to generate to constitute all blood lineages. So in this talk, I'm focused on the second 2 waves. The second 2 waves in the yolk sac, the mesodermal cell generated the homogenic cell, eventually leading to the EMP. In the AGM with potential to producing a homogenic cell, and on the path towards HSC. So the question is so what is driving such a differential sulfate choice through the same mechanism and the single cell to hematopoietic cell, hematopoietic transition EHT between AGM and the yolk sac. So toward this goal, we perform K27 active histone mark using those 2 tissues at a stage of 9.5 and 10.5. So using the Morpho algorithm for integrating the dual omics, RNA, and histone modifications, we identify the 3 classes in each data as annotated here, but we focus on the path from the endothelial cell to homogenic endothelial cell, as evidenced by the pseudo-time analysis presented by UMAP on the right hand. So in AGM, basically a single cell can be separated into 2 types of differential gene, differential expression gene, but we always with well correlated H3K27 isolation singular. and also the same to the yolk sac. So one striking thing I'd like to share with you is whether in decreasing or decreasing or increasing, the change in H3K27 acetylation signal appears to take place earlier than that in transcription. But that pattern was flipped in the yolk sac. The further examination for the enhancer linked to one gene well, the yolk sac represented a higher average enhancer number linked to one gene than that in AGM through covariant analysis. So by COTAC data further analysis, we could identify a lot of interesting transcription factors, including several known and also unknown transcription factors essential for each lineage bios, such as HOXP4, required for the pre-HSC development with higher correlation between the transcription factor promoter and the potential enhancer. So likewise, MAS1, so regular yolk sac EMP development with higher correlation between promoter and potential enhancer. So together, in the second part, I demonstrated the one example to show in how one could integrate the different omics data to understand the gene regulation, which is regulate the sulfate bowels. So as a quick summary, today I'm going to show you our new method, which is unpublished. So named COTAC for single cell detection of transcription and we are used to that then. So with COTAC, we are able to reconstruct a high-dimensional epigenome to understanding the gene regulatory relationship. In the second part, I show you two examples. How could we harness COTAC, the power of COTAC, to understanding the gene regulatory relationship leading to the sulfate bowels. So those projects are largely driven by three talented students, Haiqin, Injia, and Qian Hao, and also other member for the comments, the essential comments, and as well as the founding body. And with that, I'm happy to end here. I'm happy to take a question. Thank you.
Very interesting talk. We have a question in the chat. Dr. He, can you please explain in more detail what pseudo single cell represent from Vivian Lee?
Yeah, that's a nice question. Yeah, I think that's the advantage or the power of the COTAC we recently developed. So we just simply to measure the single cell according to the transcriptional similarity. Yeah, here's the principle.
You can type your question in the Q&A or in the chat box. We have a question from Jian Hani Kim. Do the single cell histone data also suffer from sparsity like single cell ATAC-seq? And what processing procedures are required before data analysis?
Yeah, that's quite an interesting question. Yeah, of course. Yeah, I think that like any epigenomic single cell epigenomic technology, yeah, those technologies suffering from the sparse data. And yeah, but as I mentioned, when you integrate the different omics data such as chip and transcription, they could nicely or even much pick up a rare cell type or better annotated cell type by reconstructing a multidimensional epigenome. And the second question is what sort of a processing procedure are required before data analysis? Yeah, I think it's pretty much the same as you work with single cell tech. It's pretty much the same. but yeah, there's also a subtle difference when you consider the data modality. The histone modification or the transcriptome binding, we usually producing much less rates. Transcriptome factor produce much less rates than histone modification. So keep that in mind. And so so this advantage in CoTech, they can harness the different modality for cell type annotation and genomic network understanding. So I agree with you. So nothing special, but keep in mind it's a bit different from single cell tech. So yeah, that's my short answer.
We have questions from Pu Wen. The first question is, on average, how many genes could be detected with both expression and histone modification per single cell? That's the first question.
Yeah, good question. So, so actually, when we calculate the transcription, we calculate the two ways. One way is UMI, and the second is the transcript and gene, how many genes number we detected in CoTech. So roughly about 1,000 to 3,000. It really depends on what kind of cell you are using. By using the cell line, the cell culture, yeah, it's easy to achieve about 3,000 genes detectable. And but when I think it could be this would be a general issue when you're working with the tissue, and because you never know whether the sample are handled optimally. So, but for the cheap part, histone modification or transcriptome effect, about 3,000 to 6,000 peaks or rays we could detect in CoTech. Really depends on the histone modification. We're also working on the transcriptome effect. Yeah. So yeah, I saw.
Any other questions? If not, I have a quick question. Oh, there's another one. Oops.
I saw it on the Q&A from Louis Chen. So what's the cost of a CoTech roughly? Is the protocol hard to implement compared to other protocols, such as SmartSeq2 and CellSeq2? Oh yeah, so so basically for the RNA part, pretty much similar as you work with any modified SmartSeq2 or Cell2, look at the 3' end. And the challenging part is for the single cell chip as we implemented it in the CoTech. So I don't think it's pretty hard, yeah, just pretty tricky, and the cost is pretty low. And yeah, the reason because we customize many procedure, including the sequencing strategy. We could sequence on the Illumina standard recipe, so the cost is not a big issue. So as long as you have figure out the SmartSeq2 or CellSeq platform, and from Pu Wan Tan, it's an interesting work to combine the transcriptome with the epigenome data of 2 questions. So on average, how many genes already answered? And just like other single cell method, barcode or UMI are used in your method. Yes, we did. Yeah, you referred to the paper manuscript we recently deposited at a bioarchive. Just search CoTech on bioarchive, and then you can play with it. Yeah, thank you.
I have an interesting question. You showed a very fascinating finding about the pseudo time, comparing the RNA and the methylation. I just have a naive question. Is the pseudo time computed with RNA velocity? And how do you compute the pseudo time for methylation? And,
yeah,
you know.
Yeah, I know that. Yeah, and that's an excellent question. So actually, we can do many ways. So in one way, everybody is familiar with it. You can just do the pseudo-time analysis using more like 2 or 3, or even as you just mentioned, the RNA velocity. So you rank it by transcription. However, we round up with 2 options. The first one, we could simply do the pseudo-time analysis using the epigenome data, and as also people use on the single cell attack, MONARCH3, SORAS3, and also but recently as I show you for the second part, understanding biodevelopment, we are using Morpho, the multi-omics integration factor. So basically, this algorithm consider the whole omics for clustering. So I think this could be identify, unbiased identify a cell type, or even annotated some specific cell type which have not been annotated by either omics information.
Wow,